In this video, we're going to cover spermatogenesis or sperm production. So by the end of this video, you'll understand the phases of spermatogenesis and how mature sperm cells are produced. We're also going to break down the role of supporting cells and the hormonal control of male reproductive function. So let's begin this lecture by taking a look at where spermatogenesis occurs. So spermatogenesis occurs in the seminiferous tubules, in the testes. So here's the anatomy of the male reproductive tract. Here's the penis, scrotum, testes, epididymis, and vas deferens. And this is the frontal view showing the paired structures. Now let's zoom in on the testes and briefly talk about its structure. So the testes consist of seminiferous tubules, which are a series of convoluted tubules and are supported by interstitial tissue. Now, the seminiferous tubules form the rete testes, which is a network of interconnected tubes, and developing sperm will travel from the seminiferous tubules into the rete testes, and from the rete testes to the efferent tubules, which are ducts that will transport the sperm from the rete testes to the epididymis. And in the epididymis, it's a storage reservoir for sperm. Okay, so now let's zoom in to one of these seminiferous tubules, because this is where spermatogenesis occurs. So these tubules are separated by Leydig cells, blood vessels, and nerves. So these orange cells here, these orange little guys, are Leydig cells. And we can divide the seminiferous tubule into a basal compartment, okay? So it's bound by a basement membrane. And these are smooth muscle-like cells, these here. And in the center is the lumen, the adluminal compartment. This contains mature sperm cells, spermatozoa. These compartments are divided by tight junctions, which are intercellular junctional complexes. So the basal compartment is between the basement membrane and the tight junctions, so this space here. And the adluminal compartment begins at the tight junctions and extends all the way to the lumen. Now the seminiferous tubules are lined with Sertoli cells, also known as sustentacular cells, and they rest on the basement membrane and extend all the way to the lumen. And these Sertoli cells are joined to adjacent Sertoli cells by tight junctions. Okay, so this right here is a tight junction. And the Sertoli cells form a ring called the Sertoli cell barrier or blood testes barrier around the outer circumference of the seminiferous tubule. So think of these Sertoli cells as a ring structure, okay? Because I've only drawn a part of it, but it's a ring structure. And the Sertoli cell barrier is to prevent any waste products and chemicals from entering the lumen. We want to separate it from the systemic circulation to prevent the immune system from recognizing sperm as foreign. And Sertoli cells also have other functions. They provide support to germ cells, so structural, metabolic, and functional support. They are nurse cells for spermatogenesis. They nourish the developing sperm, deliver nutrients to germ cells. They divide the tubules into compartments, and they form the Sertoli cell barrier. They also secrete fluid to form tubular lumen and ensure movement of cells within the epithelium and overall regulate the spermatogenic cycle. Now, Sertoli cells have more functions, but we're going to come back to this when we talk about the hormonal control, okay? So now let's subtract complexity and break down spermatogenesis. So the goal of spermatogenesis is to produce a genetically unique male gamete, and that male gamete can go off and potentially fertilize an ovum and produce offspring. Spermatogenesis is where a haploid cell, N, is produced from a diploid cell, 2N. It involves a series of cell phases, and we can divide it in 3 phases, and we're going to go through each of these phases. The first stage is proliferation and differentiation of spermatogonia by mitosis. So spermatogonia are undifferentiated germ cells. The second stage is meiosis, and the last stage is spermiogenesis, which is the process that turns spermatids into spermatozoa. sperm. Let's go through the first phase, the proliferation and differentiation of spermatogonia by mitosis. And we're going to come back to this diagram because I want more space to go through the overall process and simplify it. Okay, so eukaryotic organisms have different number of chromosomes in the nucleus of the cell, referring to human somatic cells, so any cell in the body except the sperm and the egg, are diploid cells. They contain 46 chromosomes, 23 from the maternal side, and 23 from the paternal side. And the number of chromosomes set can be denoted as N. So a haploid cell means there's only one set of chromosomes, or one copy. And diploid, or 2N, means it has two sets of chromosomes, one set from maternal and one set from paternal. So in mitosis, the goal is to take the genetic material, the 46 chromosomes, so the DNA that's located in the nucleus of the cell, replicate it to produce two copies, and they're identical. So here, we've produced two identical cells, and each of the two daughter cells have a full set of 46 chromosomes. And these daughter cells are going to continue to divide again and again, producing clones of spermatogonia, male germ cells. Now, let's talk about the timing of mitosis for a second, because mitosis in germ cells differ in males and females. In males, some mitosis occurs in the embryonic testes and primary spermatocytes are produced, but mitosis really begins at puberty. So now the cells from the final mitotic division are called primary spermatocytes. So this is a primary spermatocyte, and it's going to continue through the spermatogenic cycle and undergo the meiotic phase of spermatogenesis. The number of chromosomes per cell is still 46, and there are 2 chromatids per chromosome. Now, if each clone produced continues through the meiotic division, then the pool of spermatogonia would disappear. So we need a mechanism so that we can maintain this pool of undifferentiated spermatogonia. So what happens is one cell will remain a stem cell, and we call this type A. And later on, that cell can fulfill its destiny and continue on the spermatogenic process. And this one here is a type B spermatogonium, and it's going to develop into a mature sperm. So primary spermatocytes will increase in size, which is why I've drawn these cells slightly bigger. And it's going to move through the tight junctions of the Sertoli cells. Okay, so let's go back to the Sertoli cell diagram and apply this. We have a spermatogonium here in the basal compartment, and we're going to produce a primary spermatocyte by mitosis. This is happening in the basal compartment, and primary spermatocytes will increase in size, and it's going to move through the tight junctions of the Sertoli cells. So this tight junction is going to open, and they're going to enter the central compartment. And once they go through this tight junction, a new tight junction will form behind them. Okay, so the tight junction will open, and the primary spermatocyte will enter the central compartment, and as they enter, a new tight junction will form behind them. And once it's in the central compartment, it undergoes meiosis 1, the first meiotic division. That's the second stage of spermatogenesis, meiosis. So let's go back to the other diagram and break this down. So this primary spermatocyte will enter the first meiotic division. In the first meiotic division, homologous chromosomes, so these two identical sister chromatids, come together, they line up right next to each other, and crossing over occurs. So what's happening here is there is a recombination of genes on homologous chromosomes. So the sister chromatids are no longer identical because we're trying to create a genetically unique male gamete here. So these are no longer identical. Now the homologous chromosomes are lined up in the center of the cell, and their orientation is random. And then the cell divides, okay, and we produce the secondary spermatocytes. So these secondary spermatocytes contains 23 chromosomes and still has 2 chromatids per chromosomes, but they are no longer identical because of the crossing over. Now let's go back to the Sertoli cell diagram and apply this. Okay, So meiosis 1 will produce secondary spermatocytes, and these chromosomes are no longer identical because of the crossing over. Remember, the sister chromatids lining up right next to each other and crossing over occurred. Now, the secondary spermatocytes will undergo the second meiotic division. So let's go back to the other diagram and break this down. The sister chromatids of each chromosome, so both of these chromatids were originally either maternal or paternal, will separate. Again, they're going to line up in the center and they're going to separate into new daughter cells. And these daughter cells contain 23 one-chromatid chromosomes. And these are called spermatids. Okay, so we have 23 one-chromatid chromosomes. Now let's talk about the timing of the second meiotic division for a second, because the timing differs in males and females. In males, it occurs continuously after puberty, whereas in females, it doesn't occur until after fertilization, until the secondary oocyte, which is arrested in metaphase 2 of meiosis 2, is fertilized by a sperm cell. Okay, so that is the meiotic phase of spermatogenesis. Crossing over occurs and the random distribution of the maternal and paternal chromatid pairs, because our goal here is to create a genetically unique gamete. So never forget, you are unique, because the process here is creating a genetically unique gamete. There's no one else like you, one of a kind. Unless you're an identical twin, but hey, you're still remarkable. Okay, so that's the meiotic phase. Now, before we move on to the last phase of spermatogenesis, let's quickly recap what we've covered, because we've covered a lot. So spermatogonia, which are undifferentiated germ cells, begin to divide mitotically during puberty and produce primary spermatocytes. So this primary spermatocyte contains 46 two-chromatid chromosomes. Type A will remain a stem cell because we want to maintain the supply of spermatogonia. And type B, the type B spermatogonium, will develop into a mature sperm. Now, each primary spermatocyte will increase in size and will undergo the first meiotic division and produce two secondary spermatocytes. And the secondary spermatocytes contain 23 two-chromatid chromosomes. Then the secondary spermatocytes will undergo the second meiotic division to form spermatids. And each of these spermatids contain 23 one-chromatid chromosomes. So let's go back to the Sertoli cell diagram and apply the second meiotic division. So from secondary spermatocytes, spermatids are produced, and these spermatids contain 23 one-chromatid chromosomes, and this is happening in the adluminal compartment. Now, these spermatids undergoes the final phase of spermatogenesis, known as spermiogenesis. Let's go through that. So, spermiogenesis is the process of differentiation of the spermatids into spermatozoa, sperm. This involves cell remodeling, DNA compaction, and elongation. Here's the structure of a sperm. We have the head, midpiece, and tail. The head consists of the nucleus. This contains DNA, the genetic information. The tip of the nucleus is covered by the acrosome. So this is a protein-filled vesicle which contains several enzymes, and these are important in fertilization. The mitochondria forms the midpiece because the sperm needs energy for movement, needs energy for its travels. Safe travels, boys. And the tail, most of the tail is a flagellum. Group of contractile filaments that produce whip-like movements helps the sperm propel forward. And the entire process of spermatogenesis from a primary spermatocyte to a sperm takes approximately 64 days. So now let's go back to the Sertoli cell diagram and apply all of this. Okay, so from spermatids, sperm cells are produced. And once we've produced spermatozoa here, the cytoplasm of the Sertoli cell around sperm retracts, and the sperm are released into the lumen of the tubule. And this process is called spermiation. So the sperm disconnects from the germinal epithelium and now they are surrounded by luminal fluid. So that is spermatogenesis. Let's now move on to the next part of this lecture and go through the hormonal control of male reproductive functions and break down the functions of Sertoli cells and Leydig cells, the supporting cells that assist sperm development. This process is initiated through hormonal controls in the hypothalamus. So take a look at this cute and simple brain that I've drawn here, and let's zoom in to view the hypothalamus, which is a small region of the brain, and it's close to the pituitary gland. The green section is the hypothalamus, and we have the pituitary gland here. This beauty, secretes gonadotropin-releasing hormones, GnRH, and when it does that, it's going to trigger the release of luteinizing hormone, LH, and follicle-stimulating hormone, FSH, from the anterior lobe of the pituitary. Now, GnRH reaches the anterior pituitary gland via the hypothalamus-hypophyseal portal vessel. Now, something to note about these neuroendocrine cells in the hypothalamus is they fire a brief burst of action potential, secreting GnRH at these times. So during each pulse, they're triggering the release of LH and FSH, but not in equal amounts. Now, LH acts primarily on the Leydig cells, which are found in small connective tissue spaces between the tubules to stimulate testosterone secretion. So these Leydig cells here, these orange cells, are going to secrete testosterone. The testosterone secreted by the Leydig cells acts locally by diffusion from the interstitial space into the seminiferous tubules. So the testosterone enters Sertoli cells to aid sperm development. Now, FSH acts primarily on the Sertoli cells to stimulate the secretion of paracrine agents that's needed for spermatogenesis, to stimulate proliferation and differentiation of developing sperm. And Sertoli cells also secrete fluids that contain androgen-binding protein, or ABP, which binds to testosterone secreted by the Leydig cells and enters the lumen of the tubule. So ABP helps maintain the testosterone concentration in the lumen of the tubule because testosterone helps drive spermatogenesis. Now, let's go through the negative feedback effects and how the secretion rates of LH and FSH are altered. So first of all, what happens when testosterone levels are elevated? Testosterone travels to the hypothalamus and acts on the hypothalamus to decrease the amplitude of GnRH bursts. So it's going to decrease the secretion of gonadotropins. It also acts directly on the anterior pituitary gland to decrease LH and LH only. And when LH response decreases, testosterone secretion decreases because LH acts on the Leydig cells that produce and release testosterone. So this is a negative feedback loop. Now, how do we control FSH? So that is how LH secretions are altered. What about FSH? Well, when sperm production is elevated, Sertoli cells are going to secrete a protein hormone called inhibin. And inhibin is going to exert a negative feedback directly on the anterior pituitary and decrease FSH release and FSH only. And when FSH decreases, remember that FSH acts on Sertoli cells, so this is going to decrease ABP, which binds to testosterone to help maintain testosterone concentration. And testosterone is required to drive spermatogenesis. So when testosterone levels decrease, spermatogenesis decrease. And that is the hormonal control of male reproductive function and how testicular hormones exert negative feedback effects. Now, before we end this lecture, let's quickly recap what we've just covered. The hypothalamus secretes GnRH and goes to the anterior pituitary gland via the hypothalamus-hypophyseal portal vessels. The anterior pituitary gland releases luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH acts primarily on Leydig cells, which secrete testosterone, and the testosterone acts locally on Sertoli cells with the help of ABP to drive spermatogenesis. And FSH acts on Sertoli cells, which synthesizes ABP and nourishes germ cells, stimulates spermatogenesis. And when there's elevated testosterone, it will act directly on the hypothalamus and the anterior pituitary gland to decrease the release of LH and LH only. And FSH is inhibited by inhibin, which is secreted by the Sertoli cells. So that is spermatogenesis. In this lecture, we learned what spermatogenesis is and how it can be divided into three main phases. The first stage is proliferation and differentiation of spermatogonia by mitosis, the second is meiosis, and the third is spermiogenesis. We also learned the hormonal control of male reproductive function and how the hypothalamus secretes GnRH and the anterior pituitary gland releases LH and FSH. LH acts primarily on Leydig cells and FSH acts primarily on Sertoli cells. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!